Complement system. First, let me define the complement system. The complement system is a set of plasma proteins that act in a cascade to attack and kill extracellular pathogens. It approximately has 30 components: activating molecules, regulator factors, complement receptors, and membrane proteins which inhibit the lysis of host cells. The complement system is the most of the complement proteins and glycoproteins are produced in the liver in an inactive form or zygomen. Activation is induced by proteolytic cleavage. Activation of pathways. The classical pathway has antigen-antibody complexes, MB-lectin pathway has lectin binding to pathogen surfaces, and alternative pathway has pathogen surfaces, wherein it comes to the complement activation and come out as recruitment of inflammatory cells, opsonization of pathogens, and killing of pathogens. Let us now then proceed to the classical pathway. Classical pathway is a part of adaptive immune system. It relies on AgA-B complex to get activated. The classical pathway is initiated by 1. AB binding to the pathogen, and 2. C1 proteins binds to the Fc of AB. The C1 protein, C1q, has 18 polypeptides, 6 arms with globular heads. It binds Fc on IgG or IgM to get activated. 2C1R plus 2C1S are activated by activated C1q. In complement system, the antibody-antigen complex bound to C1 activates C1s, which cleaves C4 and C2 to form C4b2b or the C3 convertase. Active C3 convertase, which cleaves C3 molecules into two fragments, the C3a and C3b. C3b forms a complex with C4b2b, producing a new enzyme, the C5 convertase, which cleaves C5 to form C5a and C5b. C5b binds to C6 and C7 to form a complex, and C8 then binds to the CB5 and C6-C7 complex, followed by the polymerization of up to 16 C9 molecules to produce the membrane attack complex that generates a channel or pore in the membrane. Let's now proceed to the alternative pathway, wherein it is also called as properdin pathway. It is a part of innate immunity, an antibody independent. The alternative pathway also is slower than the classical pathway. The molecules of C3 undergo cleavage at continuous low level in normal plasma. Let me then explain the activation of alternative pathway. C3 contains an unstable thioester bond. This unstable bond makes C3 subject to slow spontaneous hydrolysis to C3b and C3a. The C3b is able to bind to foreign surface antigens. The mammalian cells contain sialic acid, which inactivates C3b. In factor B, C3b on the surface of a foreign cell binds to another plasma protein called factor B. This binding of factor B results in the exposure of a binding site for another enzyme called factor D. The binding of factor B to C3b allows a protein enzyme called factor D to cleave factor B to BA and BB. Factor BB remains bound to C3b while BA and factor D disperse away. The complex C3bBB is C3 convertase of alternative pathway. The properdin factor P stabilizes C3bBB so it may cleave more C3, may cleave over 1 million C3 molecules. It also protects BB-C3b from inactivation by complement control mechanisms. The C5 activation is the C3b-C3bBB functions as the C5 convertase in the alternative pathway. And lastly is the MAC formation. Lastly is the lectin binding pathway. It is an independent of antibodies. Lectins are proteins that bind to specific CB targets. It is activated by mannose binding lectin or MBL. MBL is a lectin that binds to mannose residues on the microbes. MBL is similar to C1q in structure and function. The activation of lectin pathway. First step is binding of mannose binding lectin or MBL to mannose residue on the surface of microbes. To the MBL bound to microbe, MBL associated serine proteases or MASP1 and MASP2 will bind. MASP1 and 2 is similar in structure and function to C1s and C1r. This complex will cleave C4 and C2. Now, let us then proceed to the effector functions of complement system. Number 1. It facilitates opsonization. It is extremely important when pathogen carries a capsule. Second is the cell lysis. And third is the immune complex clearance. Fourth, inflammatory response and chemotaxis. This is now then the summary of complement activation system from the classical pathway, alternative pathway, and lectin pathway. Next is the function of complement. First is the anaphylatoxins. C3A and C5A cause mast cell degranulation. It releases of vasoactive mediator, histamine causing anaphylaxis, allergy. Vascular changes, dilation, and increased permeability or edema. Second is chemotaxis. C5A stimulates movement of neutrophils and monocytes towards sites of antigen deposition. Third is opsonization. C3b binding to complement receptors and enhance phagocytosis by neutrophils and macrophages. Fourth is the cytolysis. The insertion of the membrane attack complex into the cell surface leads to killing or lysis of many types of cells including erythrocytes, bacteria, and tumor cells. And fifth is the clearance of circulating immune complexes. And lastly, let us now proceed to the regulation of complement system. Number 1, C1 inhibitor. It inactivate the protease activity of C1 result in inhibition of classical pathway of complement activation. 2. Factor H and factor I. The alternative pathway is regulated by binding of factor H to C3b forming complex cleaved by factor I, which result in the reduction in the amount of C5 convertase. And 3 is the decay accelerating factor or DAF. It promotes decay of C3 convertases enzymes, which is the major amplification step in complement activation result in prevent formation of membrane attack complex. That's all for my report about complement system. Thank you.